Hello, everyone. Welcome to today's webinar. I'm Alex Taylor, a contributing editor to ACS Media Group. I'll be moderating today's event, Peptide Crystallization, a Scalable Solution in Pharmaceutical Process Development. This educational webinar is sponsored by Cambrex. Cambrex is a global CDMO that develops and manufactures drug substances across the entire drug life cycle. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. A few housekeeping notes before we begin. You can adjust the size of the slides on your screen by dragging the lower right corner. If you need technical assistance, please use the help widget in the upper right corner or submit a question through the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions that you received earlier. We encourage you to participate by submitting questions at any time through the Q&A box. The questions will be answered at the end of the presentation, and as your moderator, I will pose as many as time permits. This webcast offers a certification of participation. To receive your certificate, click the Earn Certification widget on your screen to download your copy after the webinar. Please note that CNEN does not endorse any company, products, or services that may be mentioned during this webinar. A recording of this webinar will be available on the CNEN website after the live event. During the presentation, we will hear from Dr. Hannah McLachlan, a crystallization team leader with Cambrex. Hannah leads the peptide crystallization efforts at Cambrex Edinburgh. Let's get started. I'll now turn it over to you, Hannah.
Thank you very much, Alex. And hello and good morning or afternoon to everyone listening. So yes, as Alex said, I'm going to be discussing peptide crystallization, a scalable solution in process development today. So just a little bit about me. As Alex said, I'm Dr. Hannah McLachlan. I have a first-class master's in chemical engineering with pharmaceutical chemistry from High-throughput University in Edinburgh. And I then did a Ph.D. in chemical engineering with a focus on crystallization, also from High-throughput University, in conjunction with CMAC as part of Strathclyde University. So I have been at Cambrex, originally Solid Form Solutions, since 2015, and I started as a crystallization and screening scientist, which means I did a lot of the projects for both crystallization process development and screening work as well. I have been crystallization team leader since 2025, and as Alex said, we look at crystallizing peptides, but we also look at the crystallization development of small molecules, which is also a very important part. Outside of work, I like to travel, as evidenced by the lovely picture there, with me beside a giant obelisk in Egypt. I also enjoy history and reading, and I'm an enthusiastic, if amateur, crafter. So a little bit about Cambrex. So as Alex mentioned, we are a leading global contract development and manufacturing organisation and provide drug substance development and manufacturing across the entire drug life cycle, as well as comprehensive analytical services. We are based in both North America and Europe. So in North America, we have 6 process sites and our headquarters. So that includes Longmont, which is in Colorado, Charles City, Iowa, High Point in Durham in North Carolina, and Agawam and Waltham in Massachusetts. And in Europe, we have storage in Waterford, Ireland, the site I work at in Edinburgh, Scotland. So we are the solid state science and crystallization development site. More storage in Belgium, Wiesbaden, Germany, Milan, Karlskoga, and Tallinn as well. So just a little bit about Cambrex Edinburgh. We work on the Bush Estate, which is just outside of Edinburgh. So we've got a very old 18th century building on the estate. Fortunately, we do not work there. We work in the Fleming building, which is slightly within the estate and a little bit away, but has a lovely backdrop of the hills. So a quick history of Cambrex Edinburgh. So as I mentioned, we started as Solid Form Solutions back in 2008. We were acquired by Avista Pharma in 2018 and then Cambrex in 2019. And we expanded our facility in 2020 to double our original size, which has allowed us to have separate labs and office space. So what does Cambrex Edinburgh do? So we are the solid state Centre for Excellence in Crystallization. So we cover peptide crystallization screens, which we'll be discussing a bit more today, but we also look at polymorphism screens, salt and co-crystal screens, and crystallization screens of quite often small molecules, but also larger non-peptide molecules. We also look at chiral resolution screening, structural characterization, and also crystallization process development. So that looks at taking quite often what our screening team members have identified as the ideal form, and then looking to develop a process to make that on a larger scale, which can be then transferred to our production sites or other production sites as required. So our team has an excellent track record in delivering high quality science and reporting, and there are 3 team leaders, myself and 2 others, and we report to the head of solid state, and then we have multiple solid screening scientists and crystallization scientists responsible for running the experiments, analysing all their data, and compiling data packs and reports. Just a very quick look at our service offering. So overall, we have 24 staff dedicated to solid form activities. So that is our screening and crystallization staff, as well as our analytical development staff. So we have in-house analytical development who help us analyse some of our materials through things like HPLC and GC and particle size. And they also can develop methods as well. And we can cover the full range of a product life cycle from preclinical development, looking at rapid screening, right through to launch and final stage, clinical stage 3 and 4, where we can look at potentially intellectual property screens and crystallization process improvements and troubleshooting. So we'll now move on to what we're actually here to talk about today, which is peptide crystallization screening and development. So we'll start with, what is a peptide? A peptide is a short chain of amino acids, often thought of as the building blocks of proteins. Peptides are commonly between 2 and 100 amino acids in size and come in multiple different conformations, including linear, cyclic, and lasso, to name a few. So we have a couple, on the right-hand side, we have a couple of examples there. So we've got an example of a linear peptide at the top, which I've called A, and then we have an example of a potential cyclic peptide, which is B at the bottom there. Now, these are relatively short chains. They can be much bigger and much more complex. So peptide fragments are essentially short amino acid sequences which can be combined to make the full length of the peptide. So fragments can vary in length from 2 amino acids upwards and are often produced as both tagged and untagged compounds depending on the process. So there in the middle, we've got a little fragment 1 of peptide A, which, as we can see, is the first 4 blocks of linear peptide A at the top there. And it's also 4 of the blocks of cyclic peptide B. Peptides play an increasingly important role in drug discovery, with a potential to offer therapeutic agents beyond traditional hormone replacement therapies. And in addition, peptides are often also used as subcomponents in other drug substances. The molecular complexity of peptides has kept them out of development pipelines in the past, but innovations in synthesis and purification recently have supported their momentum as alternatives to potential small molecules for addressing unmet medical needs. And we have seen that quite a lot recently, and it's often in the news with the GLP-1 materials and things like that. They have other uses as well. So peptide crystallization screening. So why would you do it? So crystalline material offers many advantages over amorphous material, including improved stability and physical and chemical properties. Now, these improved properties can lead to processability improvements and enhanced handling for further development work. This means that your material might be able to be handled outside of -20, no oxygen environments, to material that actually can be handled in an open lab with much less problems. It can also be used as a purification technique, which can lead to significant cost savings compared with chromatography preparation purification techniques. So this could be purification as in your standard sense of area %, your mass on area under a curve purity, but it could also be in trying to remove some of these potential unwanted fragments that you've prepared that have sort of hung around during your preparation. Now, peptides themselves are a particularly challenging class of molecules in any crystallization process, generally due to their larger size, but also due to their flexibility. They have a high risk of gelling. They can also aggregate very easily, and fibrillation is also a problem during processing. Quite often, to try and reduce these risks, salt formation is required to obtain crystalline material or even just stable material. We have received many examples, quite a few of them are in our further in the slide deck, of receiving different salt forms to ensure you actually get a stable solid and not just a liquid or an oil. So we've got a couple of just pictures there of just crystalline material observed initially on an initial small scale screen, and then birefringent crystals, which were recovered from a slight scale up towards the bottom. So we'll now have a quick overview of a crystallization screening process. So we've got a workflow here. This is a very basic workflow, but it just kind of gives you an idea of what you might want to look at. So we have processes and conditions that are always tailored to the peptide being screened and the requirements of the project. And the experiment scale and project scope is designed based on material amounts. Peptides are quite often expensive to produce, so we know material is precious. So we have been working on and continue to work on developing process schemes that allow us to get the most amount of data from the smallest amount of material, but ensure that the data is actually applicable and accurate and not just giving false results. So from this scheme here, we can see that we always start with initial characterization just to give us a baseline of what the material is and how it works on our analytical instruments. Then the building block is always solubility, because that allows us to see how the material behaves. And then we have got three different streams there. So if we want to go down the buffer crystallization screening route, so that's looking at different buffer additives, different salts and things like that, we can look at buffer. We have to look at buffer capacity first to check how the material behaves in buffers. Essentially just to make sure that your material is not just going to degrade if we expose it to different pH systems. And then we would move into buffer crystallization screening. We also have aqueous organic crystallization screening, and we've also got salt and co-crystal screening. Sometimes we do all of these for a molecule, sometimes we do one of them. It just entirely depends, as I've said, on what the requirements are and partly how much material is available. All three always lead into scale-up trials. So this can be a very small scale-up or a much larger scale-up. And this just allows us to produce material for developability and stability assessments to ensure that what we've got is actually, well, one, reproducible, and two, is material that is actually worthwhile moving forward with, because we don't want to suggest using something that is not suitable for systems. We can then move on to crystallization process development as well, which allows us to do larger scale development, which can then be transferred to a production site. So we'll just go into a little bit more detail in a couple of the sections I've mentioned there. So the first one is research plates. So we use multi-build research plates for initial peptide screening in buffer and also solvent systems. This allows us to use very small amounts of material, so it's generally less than 1 milligram of peptide input per well. And we can screen up to 96 different conditions using different pHs, anti-solvents, buffers, salt additives, etc. at the same time. We have recently purchased an automated liquid dispenser, which allows us to dispense accurate volumes into our plates and vial systems as well, to ensure that we get accurate systems and accurate volumes repeatably throughout the process. Our experiments are always monitored initially through microscopy, so we use this to search the drop for signs of crystalline material. So we have an automated microscope, which goes up to 500 times magnification and gives us high definition images of potential particles and allows us to see whether we've got actual crystals there, also what their nature is, and also see whether we've got oils and things like that. So once we have potential crystalline particles, generally identified by polarized light microscopy, we do re-preparations on a larger scale for further analysis. So often we initially will start with a direct replication of the well plate conditions on a larger scale, so we have sufficient material for actual analysis. But we will also look at slurry trials at various conditions, so different concentrations, temperatures, to offer alternative preparation routes, because the plates themselves are very similar to vapor diffusion experiments, but looking at alternative preparation routes can be much better for actual processes. All material prepared will be analysed to determine the nature of the solid. So we look at, as well as PLM, we will look at XRPD to see if it's crystalline, and also to check whether things that are precipitated are they buffers, are they looking like they're just counter ions or components. HPLC to check for purity, as well as NMR, and thermal analysis. So this also looks to identify any potential improvements compared to the input material. So the images here are just sort of showing a sort of overview of a process that was successful. So we had a positive hit in the research plate. We then did a couple of slurry experiments, as we can see in the middle there, and we got material as a slurry. We then looked at material under the microscope again, and we had crystalline material, which was then analysed by XRPD and further systems and found to be advantageous compared to the input material. So once crystalline material has been recovered and fully analysed and scaled up to a small scale, we can then look at initial process development to identify the most appropriate conditions for preparation of crystalline material from the process. So early stage peptide crystallization development typically involves further solubility and metastable zone width assessment to identify the preferred solvent systems. So this looks at solubility of the prepared material, preferably crystalline, so we can see how that may differ from the amorphous material. And the metastable zone width allowed us to identify potential seeding zones. We then will do small-scale crystallization trials to identify a potential pathway. We can do this on a down to 1 milliliter scale right up, depending on availability of material. And we also got crystallization development to refine the developed process. Often, peptides are a little bit difficult to dry. So therefore, we have isolation and drying studies which can be completed to try and identify conditions where the material is stable and maintains its crystallinity and form and any other particle attributes, for example, maybe a water content or a salt content, through isolation and drying. And we just got some images there of crystallization process development going from an oily phase, and then after crystallization process development, going to a much more crystalline particles with a morphology that could actually be scaled and developed further for plants. So what are the differences between crystallization of fragments and full peptides? As I mentioned, fragments are part of the full peptide, and we're seeing a lot more of this as an important step, an important area of interest for various clients currently. So the crystallization of peptide fragments is an important intermediate step as it offers the possibility of purity uplift and other improvements in material properties prior to the formation of the full peptide. The crystallization of a fragment can be easier than the full peptide, just generally due to the fact that they're slightly smaller in scale, and also they often have reduced molecular complexity compared to the full peptide. If it has been shown, multiple examples in industry and in papers, that if you can increase the purity and stability of your fragment, this can then lead to improvements in your final material. So while the basis of crystallization screening for peptide fragments is the same as screening of full-length peptides, differences occur in the applied techniques. And it's been noted that, as a rule, the longer the peptide chain, quite often the longer time likely required to material to crystallize, just due to longer time for things to get into the correct formation. And during the various projects carried out at Cambrex Edinburgh, it has been observed that the same solvent system in similar conditions can be used to crystallize a fragment as well as the full peptide. However, there's always modifications to the process required. These can include, as mentioned, changes in the hold times. The concentration might need to be tweaked slightly. Solvent system component ratios and also molar equivalents of additives, etc., might need to be changed just to ensure full conversion. We've got examples of crystallization of peptide fragments and also a full molecule further on in the slides. So currently we have been looking, as I mentioned, at high-throughput peptide screening. So recent work we have been carrying out, some of it in conjunction with our colleagues at Snapdragon, has been focused on the development of high-throughput peptide crystallization screening, generally in aqueous organic systems. So the aim of this work has been to obtain initial indications of crystallization of a compound within a quick time frame in appropriate solvent systems, which then allow us to identify potential conditions for further investigation. So we've so far successfully tested at once up to 96 solvent systems with different solvent mixtures, ratios, and additives. So the solvent systems are always selected based on positive results during previous screening of other molecules. However, we do always take into account the solubility of the peptide material, because if it's very highly soluble in certain systems, it might be worth looking at alternative systems because you might not get anything out of it, which is not what we want. So the solvent systems used can be tailored to maximize the likelihood of finding quite often material, but also crystalline material. So here we've just got a couple of examples as well. And after initially dispensing the solids, the experiments are temperature cycled with mixing for approximately 24 hours, and then they're mixed at low temperature. The temperatures used are based on the stability of the material. So if your material is not stable at high temperature, we will not go to high temperature because it's not going to help anyone. And also we look at the hold times. So as I mentioned, quite often if it's a longer material, we might look at holding for a little bit longer, particularly at lower temperature. So after temperature cycling, our experiments are taken, still within our vials, and analysed by our microscope to check for signs of crystallinity. This allows us to look at the material without disturbing it too much and also without exposing it to, for example, air or anything that might disturb our particles. So any experiments showing positive results can then be separated and analysed by XRPD and HPLC for purity, depending on the material amount available. And if the material is found to be crystalline, we can also store the material at accelerated stability conditions. For example, 25 degrees Celsius, 60% RH. We have multiple stability systems in-house that we can set it up at. And the material is then checked again, generally after approximately 24 hours, to monitor for any changes or material degradations at these conditions, just to give a quick kind of overview of how the material might behave if it was to be fully developed. So I've got a couple of images there on the right. So the top one is showing some quite small biorefringent particles, but you can actually see the outline of our vial there. And then the other one is a sort of more, there's crystalline material in it, but it's within a gel. But we can see using this microscope in this system that material is within a gel, but there is something there. Because if we had not used the microscope to look in the vial, we might have just gone, oh, that's a gel. Oh, well, we'll not use that condition. But there is something there. So that system could then be something that we can look into further by, for example, changing the concentration or the solvent ratio slightly, which may reduce the gel formation and increase the formation of the crystalline material. So now we've given you a quick overview of sort of the differences in how we look at these sort of things. We'll move on to some examples. So the first example is an initial peptide crystallization screening. So we received an amorphous acetate salt, which was a peptide. It was a 34mer, which means it had 34 amino acids. And it was a dimer with cyclic branches. So that was interesting. So we had limited material. So due to the limited material availability, a very select system was looked at. So we looked initially at solubility in solvent systems that were of interest, and also a buffer capacity because the client was very interested in looking at buffer crystallization initially. We then looked at up to 168 research plates, so sitting drop trials, and potential crystalline hits were observed in multiple conditions. Using these conditions, we then carried out small-scale trials to test the initial crystallization conditions indicated by the research plates, and then prepare further material for analysis. So crystalline solids were recovered. However, it was found that they actually weren't very stable under storage, even at -20. A further batch material was received, and at the client's request, we looked at salt swap trials. So they had specific salts, which was a hydrochloride and a sulfate they wanted us to look for, because these were of interest to them for development. So we carried out specific salt trials. So the HCl salt was successfully prepared, and then we used this for further sitting drop crystallization screening experiments. From these experiments, crystalline material was recovered and re-prepared on a larger scale for further testing, and the material recovered from this was found to show a purity uplift and be stable under selected conditions. So this was a really positive result for the client. So we can see there, we've got sort of a couple of examples also of crystalline material that was recovered from the sitting drop, and then also from the small-scale trial. A monomer fragment of the same molecule, so that was 17mer, so essentially half, was also tested, and we had similar results actually. And we did salt screening from this quite early on because this was received a little bit later. And the potential HCl salt was identified again using similar experimental conditions to what we had for the dimer. So from a different project, we received an amorphous HCl salt. This one was linear. It was a little bit smaller. It was only a 16mer. And it was again received initially for peptide crystallization screening. Again, initial material looked at solubility, buffer capacity, and sitting drop trials. However, in this case, from our solubility studies, we actually had two potential crystalline forms which are actually patterns 1 and 2 over there, from initial screening. We had limited hits from the city and drop trials using the buffer systems, and actually most of the solids recovered were subsequently identified as buffer mixtures or just buffer components themselves. We then looked to aqueous organic crystallization trials, initially looking at those 2 solvent systems, which are very positive, and then also other solvent systems on top of that. So we looked at different techniques, including temperature cycling, anti-solvent addition, slow cooling, and evaporation techniques. So from these trials, we recovered a further 3 crystalline patterns, 3, 4, and 5, in the XRPD diffractograms there. And upon drying, they were all identified to go to the same form, which was pattern 3. So we looked to scale up pattern 3, and this indicated the material was actually likely a hydrated form, which was, the client was fine with that. So initial amorphous material that we received was not chemically stable at ambient conditions and had to be stored at -20, so much so that even if you had to only take out of the freezer in small batches because there was potential that it was just going to degrade quite quickly. However, after crystallization and the screening, the pattern 3 material showed good physical and chemical stability up to 1 month at stress conditions. So we can see from the 2 impurity tables at the bottom there, the one on the left is the purity of the crystalline material which we received, we prepared, so this was pattern 3. This is our stability input, and it's got purity of about 99.1% area there. And after 4 weeks at 25 degrees Celsius, 60% RH, the purity is stable. It's actually gone up very slightly to 99.2% area, but we noticed this as well for the storage at 4075. It didn't go up, it just stayed the same, but essentially they were staying quite consistent. So the client then requested that we do some initial crystallization process assessment for the peptide. So as mentioned, we've got pattern 3, and we looked at initial crystallization process assessment, and we received 20 grams of material to carry this out. So initially we used 20 ml vials with overhead stirring. So we've got those, the image there on the far right, the vials with stirrers. And then we transferred the system into an EasyMax system, so our 102, so our 100 ml system, with our Blazemetrics probes added so we could monitor the crystallization process. We identified a process that was suitable, and we then prepared a larger scale batch, so it's about 5 grams. As I mentioned, we only had 20 to begin with. And the crystalline material was used for initial pre-formulation and bioavailability studies, also carried out at Cambrex Edinburgh, to give an idea of whether a material could go through into a cell successfully. So we had some trials were faced during this crystallization process assessment, and we just got a little table here to sort of discuss them. So one of our initial problems was long filtration times due to small particles. So we looked at varying things like the cooling rate and the anti-solvent addition rates to improve the crystallization control and prepare bigger particles, which therefore reduced the filtration times. We also did note that sometimes, partly due to the long filtration time, we were actually getting gel formation beginning to form on the top of the material, which was then slowing down filtration even further. So it was observed that we added a nitrogen blanket to the filtration setup initially before we looked at the particle size. This did help reduce the risk of gel formation. We also had relatively low isolated yields. So we looked at trying different solvent ratios, temperatures, and aging periods, and identified the optimal conditions for higher yields. And on initial trials, we did observe a slightly high residual solvent content. So this means our residual solvent content, which in this case I think was 2-propanol, was higher than the ICH limits. So we looked at drying studies, and we managed to identify suitable conditions to maintain a high purity solid, but we managed to use our humidifier drying setup to give us low residual solvent levels, so well within the ICH limits. Well, I think they were all well below 1,000 ppm, while maintaining the appropriate water content, because this, as we mentioned, was a hydrated form. So this initial development will provide the route to isolate material with suitable crystal properties for further development studies. We've got another example here. So this was a salt screening assessment of a peptide. So again, we had an acetate salt with a linear peptide. And this was to be carried out through both traditional screening processes and also using research plates as a test. So initial small-scale trials were carried out using 8 counter ions with either 1.1 or 2.2 equivalents of acid added with dissolution at 20 degrees Celsius and precipitation through anti-solvent addition and cooling. So from this, crystalline salts were recovered from, again, hydrochloride salt, methane sulfonic acid, and L-tartaric acid. An NMR analysis of the experiments indicated that potential salts had been formed. Using the research plates, materials prepared is using stock solutions. So we had a prepared solution of the peptide and water with the addition of 0.5 molar acid stock solutions, adding to both the posts, and then we had anti-solvent water systems added to the wells surrounding the posts to give us a vapor diffusion style experiment. Again, we used the same 8 counter-ion systems and looked at varying water ratios and concentrations as a pre-designed matrix based on the initial results. So after 1 day, we had precipitation observed from 16 experiments. And after a week, we had crystalline material in the phosphoric acid experiment with 2-propanol water. We did also have oiling observed in 1 experiment, which was unexpected based on the previous results. So from this experiment, we then moved on and we looked at 4 potential salt systems for further assessment. So initial trials identified a salt break procedure to form the free form from the hemiacetate salt input which was received. This procedure was successful on a variety of scales. We looked at very small scale. So initially it would be done on about 25 to 50 megs. And then we scaled this up to 500 meg, 1 gram, 5 grams and 10 grams. Excuse me. We also then looked at initial small scale trials completed to attempt to prepare multiple crystalline salts for further testing. From these experiments, we found 4 different patterns of an HCl salt, and methane sulfonic and the L-tartaric acid salts were again successfully formed as crystalline materials. Based on the analysis of the material formed, we had a bis HCl salt was selected for crystallization development. This was pattern 4. And further solubility work was then carried out on the target salt form, followed by mid-stable zone with determination in the preferred solvent systems. Crystallization development was completed, and it was identified that propanol water systems were good for the crystallization. So this was actually found to be successful in both 2-propanol and 1-propanol water, with very similar results from both systems. So the crystalline material was recovered and maintained on drying, and a high purity solids of about 98.2% area was recovered, and the input was about 95.5. So again, we've got the impurity tables here. So the one at the top is after crystallization, and then the one at the bottom is the input material, and we can see that the number of impurities has just reduced quite significantly. So we then move on to another experiment. So this one is looking at both full peptide and fragment crystallization. So we have a client who has a relatively large linear peptide. It was a 46mer, which had been isolated as a TFA salt through lyophilization. This was the only way that they had found to get solid out of their system. Crystallization screening of this material produced crystalline material after reverse anti-solvent addition in a dilute system. So this is an operating concentration of about 5 milligrams per milliliter. So prior to discovering this reverse anti-solvent addition method, we had done a large program of sort of anti-solvent cooling experiments, buffer crystallization screening, salt preparations. It was a fairly extensive crystallization process development and crystallization screening process looked at. However, this material that was crystalline, we can see there our material there from our reverse anti-solvent addition in our image. There was really quite low yields noted. And in fact, I think that particle is probably the only thing actually in the vessel. We looked at holding for longer time at low temperature after everything had been added to attempt to increase the yield. However, this led to the formation of a gum or gel-like material around the stirrer. We then carried out further trials, looking slightly at different solvent systems, so just tweaking the solvent system slightly around what we had initially used. Again, we observed crystalline material immediately upon formation, but as soon as we tried to isolate this material, it just led to the formation of a gum or gel. This was not ideal for further development. We did find another crystalline material from the same input, sorry. And this could be recovered from slow evaporation of the peptide in water. However, this did take place over 1 month. We did note a drop in purity also with the material, and this was just due to the prolonged time in solution. Again, we looked for, as I mentioned, alternative salts. As this was a TFA salt input, we did have to look at We did try to do traditional salt breaking, but that was not successful. But using resin salt swap experiments, we were successfully able to remove the TFA salt and form an HCl and an acetate form. No stable crystalline forms of the HCl salt could be identified during the further screening of this material, and a potential crystalline form of the acetate salt was identified from DCM. However, on scale-up preparation of this scale-up, this material was not successfully recovered, and again, a gel was formed. As this was a full, relatively large peptide, we were asked to look at the fragments of this material for crystallization and salt formation. So in total, 6 different fragments of that 46mer were screened at varying sizes. So I think our smallest was 4 amino acids, and our largest was about 24. So 5 out of the 6 were successfully crystallized during the screening experiments. There was one that did not want to crystallize. That was a 22 amino acid material. It was quite a long chain, and it was relatively linear, so there was no points where it could connect to itself, and we just seemed to get gels or gums again through our extensive screening program. During our screening, positive results were actually shown from the received free base material of the 24mer fragment. The client then identified a potential TFA salt of this fragment, and they wished to investigate this further. So again, we looked at screening of this TFA form, completed using the most promising methods identified from the screen of the free base. And from this, we actually recovered crystalline solids from 3 different DMSO solvent systems. So DMSO water, DMSO acetonitrile, and DMSO ethanol. And all 3 had a high purity. And the client was very happy that we had shown that this system could work for their TFA material and also their free base, depending on the route they wanted to go forward with. So another example from the same group of work, we had one of our peptide fragments was a waxy gum-like material. It was actually relatively small. It was an 8mer, but it did have, partly due to the gum-like nature, a 60% area purity. We did solubility screening on this, and we got white solids from that, which were crystalline by XRPD, and this was recovered from 15 of the 22 solvent systems tested. However, there was indications that there was a relatively low yield was recovered. Screening was carried out, and multiple crystalline forms were identified. And on drying, these were determined to be similar to the input material, which is form 1, with an improved crystallinity and purity. So we've got pattern 1 in the XRPD fractogram stack there at the top. So the received material had that pica around 18 degrees 2 theta, and then just a very large amorphous hump. So we could get a crystalline form of that. When I say they were similar, that means there was some shifts, a couple of additional peaks, but that when drying, they all sort of became pattern 1. Which suggests to us that these might be material form changes or differences due to solvent or water. However, from this pattern 1 material, the recovered yield was still identified as a potential issue, with low solids recovery observed. Again, we looked at salt screening of the material, and we recovered 3 separate highly crystalline patterns with an up to 35% area increase in purity. So pattern 1 from the salts was identified from multiple counterions in acetonitrile, and further analysis showed that this was actually the freeform pattern 1, and we had an improved yield to the previous pattern 1, likely due to the acidic environment that we were preparing it in. And again, the purity was still very high in these. We also had pattern 2, which was a sulfate salt, which on extensive drying studies formed an anhydrous crystalline form, which is pattern 3 there. So we've got 1, 2, and 3. And the third initial form from the salt screening was a Cam410 sulfonic acid salt, but the client did not wish us to look into that any further. So we'll now move on to further peptide fragment crystallization. So this is a sort of overview of our recent high throughput work. So we've looked at crystallization screening using the high throughput screening setup. This has been completed so far on approximately 18 peptide fragments, which are ranging in size from 3 amino acids to 13 amino acids in length. Material is often dissolved in a dispersion solvent, and then once added to the 96 vials, solvent is removed through fast evaporation. 95 different solvent systems for the examples we have looked at have been tested at once, and we've been keeping one blank as a vial to allow us to compare what material looks like and also the purity after it's been in the dispersion solvent. Positive results have been obtained from all the fragments we have tested so far, with crystalline or partially crystalline material covered by PLM and confirmed by XRPD, with a purity uplift also observed for many of these samples. So an example of results for a 5-mer with an initial purity of about 73% area is shown in the table there. So we've also got examples of some of the images taken on the next slide. So we can see that we've got a wide range of experiments. So we've got some neat alcohols here, variety of solvent mixtures with water, brine, heptane, TBME, and then also our blank. So we're getting mixtures of some crystals in our gel from the methanol there. A thick slurry, which means that we couldn't actually necessarily see through the image to get clear pictures. But this was also, if we've got a thick slurry, we will separate it and look at it by XRPD. And in this case, it was crystalline. And we've also got gel and some agglomerates. So this gives sort of a range of what we can see. We'll go on to the next slide and sort of look at the images. So we can see we've got some crystalline, some purely crystalline, and some amorphous materials there, and a purity there. So as I said, about 73% purity in the blank there. And then we've got up to 93% purity there. So quite about 20% purity uplift in that case. Any systems which have been given positive results are then re-prepared on a larger scale. And again, further process improvements and systems are tested around that to give us the best system to move forward. But this allows us to look at a relatively small scale and screen for a large number of potential solvent systems in a relatively quick time. So we're going to look at some of the images. So as mentioned, methanol gave us a gel. We can see from the image on the top left there that is gel-like. The middle one on the top row there is ethanol water. We can see we've got small biorefringent particles. And then ethanol brine, also small biorefringent particles, and both of them were crystalline by XRPD. The 1-propanol aqueous lithium chloride, if I just scoot back, number 19, gave us crystals. They were purely crystalline by XRPD, but we can see we've got crystals there. DMF water gave us fairly agglomerated particles, and then as did 71, which is 1-methoxy-2-propanol water, again giving us nice biorefringent particles. So the advantage of the microscope we've been using is that we can actually get quite close if need be to see and confirm that the material we're looking at is how it looks and if it's crystalline or a gel. And we can also look at both cross-polarized, which the images are shown here, and plain polarized light, so we can see it with and without polarization added to see the different effects. And our final example set, this was again a full peptide and fragment crystallization. So we had a large dimer peptide material, again, fairly big this one, and it had a PEG moiety in the middle as the linker. And we did looking for screening again for both crystallization and salt. So extensive crystallization screening was carried out, and salt screening as well. And from the crystallization screening in DMAC acetonitrile, we actually got small crystalline particles, which we could see by PLM. So we've got the images at the bottom there. So if we can see, they're actually very small particles, because even the image on the far right, is by 20 times magnification. So they're actually very small particles, which we could see by PLM. They're biorefringent. However, if we look at the XRPD, so this is the wet and the dry, which is the XRPD stack shown there. There's limited crystallinity, but you'd almost say they were amorphous. But we did see impurity uplift noted for these solvent systems, but the material was slurried for a long time, so I think up to 21 days. We also looked at doing it with sort of small overhead stirrers in our Crystal 16. We also looked at shaking it, so not magnetically or manually stirring it. Unfortunately, the crystals would just not grow any bigger than what we were seeing in these images and for this particular material. However, we also got the monomer from that material, still with the PEG moiety as one end, and this was received again as a TFA salt. So we looked at both crystallization and salt screening. So we looked at initial screening with a focus on the solvent systems, which had shown promising results from the dimer material. We also looked at other salt systems and techniques, but we found that quite often using that as a sort of basis for what might work can be successful. We also did buffer crystallization screening, and from this one, we actually had 3 different crystalline forms were identified from the buffer-based systems. 2 of these were later shown to be either buffer components, including PEG, or just the buffer itself. However, we did have 1 promising crystalline form, which had a purity uplift, and this was observed from a tris buffer system with magnesium formate. Further analysis of this material showed that the recovered solid was a formate salt, we know this because we had a significant reduction in the TFA content and an increase in the formic acid content, and there was no residual solvent present. So the results were confirmed by both HPLC organic acid and 1H-NMR analysis of this material. We have noted sometimes, depending on the peptide material, that when we go to do HPLC organic acids with the TFA, we get some overlap of some of the peaks. So that's why we do both, just to make sure that we know what is going on with the system. So in that case, it was actually a 19F fluorine NMR carried out as well. So we also had a further crystalline form out with the buffer system from DMAC acetonitrile again. And this had a high crystallinity and a good purity uplift. So analysis indicated this material was a potential DMA solvate. We also noted differences in the XRPD patterns at different solvent ratios. So here in the table, or in the XRPD diffractograms, we have pattern 4 as the top two, because we looked at both seeding and non-seeding, and pattern 5, and then pattern 4 again. So 4 was initial what we found. We then found pattern 5 during a mini scale-up. And then we looked at trying to shorten the length of time and adding seeding, which is why we've got the top two diffractograms there. And again, they're all very similar, but there are shifts. Analysis of these by, it was NMR to get solvent content, looks like there's shifts in the amount of DMA that we're actually seeing. There's no acetonitrile by NMR, but it looks like it's the amount of DMA that, depending on how well it's been dried, we're getting different slight changes in the XRPD diffractograms. And as we can note, this is actually the same solvent system that gave us the small crystalline particles for the dimer. So looks like this particular peptide sequence quite likes DMA acetonitrile. As I mentioned, we also looked at salt screening on the monomer. This used resins first to remove the TFA, and then we looked at different resin systems. So again, we got a chloride-based resin, which produced a chloride salt with a very low TFA content, and using the acetate resin in this experiment, removed the TFA and actually formed a free form of the material, which the client did not know could exist. So salt screening using both acidic and basic counterions due to the pKa's of the material was then completed, as well as crystallization screening on the chloride material. So we used the free form as input to the salt screening in this case. We had one potential crystalline chloride form from modified antisolvent addition on a small scale. However, on scale up, a gel formed, which was not what we wanted. From the acid systems, we only got gels, and any solids recovered from evaporation were amorphous by XRPD. And from the basic counterions, we did get solids from everything. However, some of these were recovered after evaporation. And any crystalline peaks noted in the XRPDs for these were identified as counterion, and a purity decrease was actually noted in 7 of the 9 solids analysed, indicating that the basic conditions for preparation of these solids was actually slowly degrading the material. Sorry. So as a final part, and to finish off this full peptide and fragment crystallization story for this set of materials, for the monomer, we looked after the crystallization and salt screening. We had 4 materials selected for scale up and then also stability studies just to see how they all worked. So we looked at the formate salt, which was pattern 2 initially, and this was successfully reproduced on a 250 mg scale with the similar purity uplift that we'd seen on the small scale. Stability analysis material showed that material remained stable over 1 month at 25 degrees Celsius, 60% RH. So we can see there at the top in number 1 in that table there, pattern 2, 97.4% area in, and after 4 weeks, 97.2. So very similar and no change in XRPD pattern. The TFA salt, which is the DMA solvate, so that's pattern 4, successfully reproduced on a 100 milligram scale. And as I mentioned, we had slight variations in the diffractogram depending on the DMA content. And this was noted depending on how long we mixed it and also our drying. So pattern 5 was identified, which looks like it's the experiment where more of the DMA has been removed. Stability studies on this material showed a loss in crystallinity and purity over 2 weeks at all storage conditions tested. So we can see there from number 2 in the table, pattern 4, 94.2% area in, and after 2 weeks, even at 2560, it's now purely crystalline pattern 4, and we've dropped down to 89.45, and that's the best purity we had from those samples. We also looked at the prepared chloride salt and the free material. These were both amorphous from the resin salt swaps. They didn't show any crystallinity or storage, and we actually had a drop in purity, particularly observed to a higher scale for the free material. The purity of the chloride salt did drop after 2 weeks, 50 degrees Celsius, but remained consistent with the 2-week result generally after 4 weeks. However, both of these materials show quite a significant drop in purity under humid conditions, particularly if you're looking at elevated temperature. The stability of the received amorphous TFA material was also studied just to see how that did compared to the other two amorphous salt, or the other amorphous salt and the amorphous free material, essentially. And again, this showed purity drops, and again, particularly under humid conditions. So we've just sort of got some examples in the table there. So the amorphous free material, 92.5 to 91.7% area after 4 weeks, 2560. And that was actually the best result we got for that material. The other two conditions showed significant degradation. The amorphous chloride input, 96.1. So after 2 weeks at 50 degrees Celsius, got down to about 95.5. And after 4 weeks, this was 95.2. So very slight difference there. And the received amorphous TFA salt, 92.3 input. And again, we've got 88.6 and then 90.9, which was our better purity there. So that is me. Yes. So that was a very overview and some examples of our peptide crystallization screening work. So thank you very much.
All right, thank you so much, Hannah, for that fascinating presentation. And now we will open it up for questions. And as a reminder to our audience, you can continue submitting questions through the Q&A box. So our first question is, how does the research plate setup work? Is it only vapor diffusion?
Yeah, so essentially it's a mixture depending on what you've got your component is. So if I go back to my picture, and sorry. So in the image here on the left, we have a small post in the middle of our well, and surrounding it entirely is our well, essentially. So to that, we add a small 1 part of sample, generally as a solution, and 1 part of our reagent. We start off with equal volumes, but again, we can change ratios depending. And then surrounding that, we add the reagent just to give us an environment. So we seal the plate, and this gives us a saturated environment. So if there's any volatile components, you will get vapor diffusion between the two, but we will also get direct contact in the well. So in the post. So the well essentially gives us a saturated environment to ensure that nothing else gets in and affects this material.
Mm-hmm. All right. What scale can you do initial crystallization process development on?
So again, we can tailor this to material availability, but the smallest scale that we would do sort of experiments that are actually applicable would be our 20 ml vial. So this is with our overhead stirring. We can do 50 right up to 5 litres on site. And then of course, some of our other sister sites can do much bigger than that. So we can do initial on a very small scale, and then it can be scaled up depending on material availability.
Oh, excellent. For the high throughput, do you need solubility information for all of the solvent systems to be tested?
So no, I mean, the more solubility data available is always better, but we can just look at selected solvent systems that are most relevant, and then we can use some modeling. We can also from there infer what similar solvent systems might behave in a similar way.
You had mentioned on the slide discussing why do peptide crystallization screening that peptides aggregate quite easily. Would you say that this is true for all of the possible peptide morphologies, including linear, lasso, and cyclic?
From what we have seen, yeah, they quite like to, all of them seem to aggregate quite freely, but it depends entirely on the material itself and how it's going to behave. So generally from what we have seen, they will aggregate, but it all depends on the different systems.
What minimum amount of peptide is needed for salt screening, and what purity, say for a 40 AA peptide?
I mean, in terms of purity, generally the higher the better, because you're going to get more, you're going to get potentially more out because there's less impurities, which could be inhibiting things. In terms of amount, as said, we looked at very small scale. So I think I mentioned that we did experiments where we looked at research plate scale. So that was about 1 gram for 9. Oh no, sorry, that doesn't work. 100 meg for 96 experiments, but it just depends. We would always design a system that is most appropriate. Ideally, you want a couple of grams if possible, but even just one, an initial screen to give ideas of what's possible can be done. We have a work program that looks at screening on a very small scale with selected systems on a 1 gram scale.
Mm-hmm. All right. Can a 20 to 30 amino acid peptide that is highly hydrophobic be crystallized? And what is the best approach to crystallize it?
I mean, potentially, without actually seeing the actual material itself and knowing how it behaves, it's very difficult to tell. It can be done. We have seen it done in the past. It just depends entirely on sort of structure and the makeup of the material, because some of them will work very easily and some of them it might take more, but might be a way to get something out of it. But as I said, it's very difficult to say without actually, seeing the data and the sort of structure and things like that.
Mm-hmm. In case you have several polymorphs, how do you get to know which one is the thermodynamically stable one?
So we look at things like, we always will run, as I said, thermal analysis, so TGDSC, to see if there's a change in different melt. We can also look at stability. So as I mentioned, we've got varying different stability conditions we can set things at as a sort of final sort of setup. But mainly through thermal analysis, we've got standalone DSCs, TGDSCs, we've also got a VT XRPD, which can look at variable temperature, X-ray powder diffraction, to see if there's any differences if you expose it to temperature holds for shorter or longer times.
In the high throughput peptide screen, are you able to collect high throughput X-ray powder diffraction of the wet cake samples?
Yes. Sorry, we can do that. Yeah. So as I said, generally we will look initially at PLM just because that is quite a quick technique and it allows us to see whether things are likely to be, whether material looks crystalline or it looks like a gel. But we have high throughput XRPDs. We have 2 that function, and we can look at our maximum plate size is 96 well plates actually, so we can look at them straight if need be.
All right, and this will be our last question today. Given an amorphous salt for the salt screen, do you need to freebase it first and then add acid, or add acid directly to the amorphous salts?
We generally try both. Well, it depends what your if it's a salt that you're receiving, it depends what your counterion is. If it's a strong counterion like a hydrochloride or a TFA salt, that is unlikely to come off if you just add another acid in. That's probably going to be the preferred salt. So we would look at potentially salt break through either traditional salt break methods or resin-based systems. But if it's a less strongly attached salt, for example, an acetate, we might look at seeing whether we could add in a stronger counterion and kick off the acetate, but keep the other counterion there. So we look at a mixture of methods to determine which is the best one for this process without causing too much degradation or other issues that we wouldn't want to see.
Mm-hmm. Excellent. Well, that concludes today's webinar. Thank you again, Hannah, for that excellent presentation. And any questions that we didn't get to during the webinar will be answered by email. Thank you to our audience for joining us, and to On24 for their technology support and production services. And a special thank you to Cambrex for making this event possible. Be sure to check the CNEN website for information on upcoming webinars. And for CNEN webinars, I'm Alex Taylor. Goodbye.
Thank you.